Hi, I am Vendrila Bonik and I am back with the part 4 of carbohydrate metabolism, that is gluconeogenesis. So from the name it is very much clear that it is the process of synthesizing glucose from some precursor. So gluconeogenesis is the formation of glucose from the precursors that are not carbohydrate, and it occurs mainly in liver during overnight fasting, and during prolonged fasting it occurs mainly in kidneys. The main precursors for gluconeogenesis they are lactate, amino acids and glycerol. So these three are non-carbohydrate derivatives. Gluconeogenesis involves several enzymatic steps which do not occur in glycolysis. Thus, glucose is not generated by simple reversal of glycolysis. So, there are three such regulatory steps in gluconeogenesis which are not the reverse of glycolysis. Apart from this, other reactions are reversible in nature. So let's discuss about the precursors of gluconeogenesis. First one is lactate. So as we know, lactate is derived from pyruvate from anaerobic glycolysis by the enzyme lactate dehydrogenase by the exercising muscles or those cells which lack mitochondria. The lactate which is released into the blood is taken up by the liver, and in the liver this lactate is converted into pyruvate by the oxidation reaction, and the pyruvate that is formed is then leading to the formation of glucose which is released back into the circulation. So pyruvate is giving rise to lactate in anaerobic glycolysis by lactate dehydrogenase which is releasing into the blood. This lactate is taken up by the liver and it is converted into pyruvate in the Cori cycle. Now next is the amino acids. We know amino acids are formed from the degradation of proteins. So the amino acids which are produced by the hydrolysis of the tissue proteins, they are the main source of glucose during fast. Their metabolism give rise to keto acids which in turn give rise to pyruvate. This pyruvate is either converted to glucose by gluconeogenesis or alpha ketoglutarate which produces oxaloacetate by transamination reaction. This oxaloacetate that is produced is the direct precursors for phosphopyruvate. So amino acids which is produced from the hydrolysis of tissue protein is giving rise to alpha keto acids. These alpha keto acids are giving rise to pyruvate. Pyruvate is converted to glucose or pyruvate is converted to alpha ketoglutarate which is the intermediate of TCA cycle. This alpha ketoglutarate is transaminated to oxaloacetate. This is again another intermediate of TCA cycle. This oxaloacetate finally is converted into phosphoenol pyruvate. Note one thing, that the acetyl coenzyme compounds or other compounds which give rise to acetyl CoA like leucine, lysine and acetoacetate, they cannot synthesize glucose because the conversion of pyruvate to acetyl CoA is an irreversible reaction. That means the reaction of the enzyme, that is the pyruvate dehydrogenase complex, it is irreversible in nature. Thus, we will not get pyruvate from acetyl CoA. So, the acetyl CoA will be forming ketone bodies instead. Next precursor of gluconeogenesis is glycerol. Glycerol is released from the hydrolysis of triacylglycerides in the adipose tissue, and this is delivered by the blood to the liver. Liver phosphorylates glycerol to glycerol 3-phosphate by the enzyme glycerol kinase. This glycerol 3-phosphate is oxidized to dihydroxyacetone phosphate by the enzyme glycerol 3-phosphate dehydrogenase, and this dihydroxyacetone phosphate is another intermediate of glycolysis. So the conclusion is that lactate, which is produced from anaerobic glycolysis from pyruvate, is taken up by the liver and it is converted into pyruvate in the Cori cycle. Pyruvate is then forming glucose, which is released back into the circulation. Amino acid, which are produced from the proteins, they are giving rise to keto acids that in turn give rise to pyruvate or alpha ketoglutarate. Pyruvate converted into glucose by gluconeogenesis, or alpha ketoglutarate, which is an intermediate of TCA cycle, is converted into oxaloacetate by transamination reaction. This oxaloacetate then finally converted into phosphoenol pyruvate. And lastly, glycerol converted into glycerol 3-phosphate in the liver, and glycerol 3-phosphate converted to an intermediate of glycolysis, that is dihydroxyacetone by the enzyme glycerol 3-phosphate dehydrogenase. Note one thing, that all the oxidation that are taking place, they require the enzyme dehydrogenase. So this enzyme dehydrogenase is involved in oxidation reduction reactions, and this give rise to NADH or FADH2. Now let's discuss the reactions of gluconeogenesis. Before starting this reaction, let me first tell you one thing. In glycolysis, there were 3 steps which were irreversible in nature. First one is the conversion of glucose to glucose 6-phosphate using an enzyme hexokinase. Next is the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate by the enzyme phosphofructokinase 1. And lastly, phosphoenol pyruvate is converted to pyruvate by the enzyme pyruvate kinase, and all these 3 steps are irreversible in nature. So these steps cannot be easily converted during gluconeogenesis pathway. So these 3 steps are the regulatory steps of glycolysis, which is also the regulatory steps for gluconeogenesis. So in gluconeogenesis, these reactions require some other enzymes for the conversion. The first one is the conversion of pyruvate to phosphoenol pyruvate, because phosphoenol pyruvate is converted to pyruvate by the enzyme pyruvate kinase, which is irreversible in nature. So we will not get phosphoenol pyruvate directly from pyruvate. So in the liver, pyruvate is converted to phosphoenol pyruvate indirectly through oxaloacetate. So the pyruvate kinase is involved in irreversible conversion of phosphoenol pyruvate to pyruvate. Now, how is this achieved? Pyruvate is first carboxylated to oxaloacetate by pyruvate carboxylase, and this requires the cofactor biotin, that is vitamin H. So pyruvate is converted into oxaloacetate, but the problem with oxaloacetate is that oxaloacetate cannot pass the mitochondrial membrane. So, but the glycolysis pathway is occurring in the cytosol, and the oxaloacetate that is formed is in mitochondria, but oxaloacetate cannot pass through the mitochondrial membrane into the cytosol. So for this reason, a shuttle mechanism is required for the transportation of oxaloacetate into the cytosol, and that is malate's oxaloacetate malate shuttle. Oxaloacetate cannot pass the mitochondrial membrane, so it is first converted into malate by mitochondrial malate dehydrogenase. The malate then enters into the cytosol, where it is converted into oxaloacetate by cytosolic malate dehydrogenase. So note one thing, the conversion of oxaloacetate to malate and malate to oxaloacetate requires the same enzyme, malate dehydrogenase, but their locations are different. So when oxaloacetate is converted to malate in the mitochondrial, the malate dehydrogenase is the mitochondrial malate dehydrogenase. On the other hand, malate, when converted into oxaloacetate into the cytosol, there it is cytosolic malate dehydrogenase enzyme producing NADH. Next, the oxaloacetate, which is now present in the cytosol, is decarboxylated to phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxykinase, and this reaction is driven by the hydrolysis of ATP. So we got phosphoenol pyruvate from pyruvate via oxaloacetate. Now the pyruvate that we got then is acted by the reactions of glycolysis, which runs in the reverse direction till fructose 1,6 bisphosphate, because we know fructose 6-phosphate, the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate is irreversible in nature by the enzyme phosphofructokinase 1. So this phosphofructokinase 1 is irreversible in nature. So this enzyme cannot be utilized to convert fructose 1,6 bisphosphate to fructose 6-phosphate. So we need some other enzyme to act on fructose 1,6 bisphosphate so that it is converted into fructose 6-phosphate, and that is the second regulatory reactions of gluconeogenesis. So let's see this. You see, pyruvate, it is converted into oxaloacetate. This is the pyruvate converted into oxaloacetate using carbon dioxide. This oxaloacetate traveled into the cytosol via malate, where it is converted into phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxykinase, that is PIPCK. This phosphoenol pyruvate is now undergoing reversible reaction till fructose 6-phosphate. You can see these all are reversible reaction, but you see the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate is irreversible in nature. So another enzyme, that is enzyme 3, is required for the transformation of fructose 1,6 bisphosphate to fructose 6-phosphate. So let's see the second conversion, that is the conversion of fructose 1,6 bisphosphate to fructose 6-phosphate. This is done by the hydrolysis of fructose 1,6 bisphosphate by fructose 1,6 bisphosphatase, which is found in liver and kidneys, and this bypasses the irreversible reaction of PFK1 of that reaction of glycolysis, that is PFK1 when it is transforming fructose 6-phosphate to fructose 1,6 bisphosphate, and the fructose 1,6 bisphosphatase forms fructose 6-phosphate. So what happened? Fructose 1,6 bisphosphate is converted to fructose 6-phosphate by the enzyme fructose 1,6 bisphosphatase, because the enzyme PFK1 cannot convert this fructose 1,6 bisphosphate to fructose 6-phosphate due to its irreversible nature. High AMP ATP ratio signals that the cell is in low energy state, so it needs glucose, so thus it inhibits fructose 1,6 bisphosphatase. But low AMP and high ATP value, it will favor gluconeogenesis. Another thing is that fructose 2,6-bisphosphate inhibit allosterically the enzyme fructose 1,6-bisphosphatase, which is regulated by insulin-glucagon ratio. And note, this fructose 2,6-bisphosphate is produced from fructose 6-phosphate by the enzyme phosphofructokinase 2. So, till here what happened is that pyruvate is converted to phosphoenol pyruvate via oxaloacetate. This oxaloacetate is then converted into phosphoenol pyruvate. Phosphoenol pyruvate undergoes several reversible reactions in glycolysis and forms fructose 1,6-bisphosphate. But this fructose 1,6-bisphosphate cannot be converted into fructose 6-phosphate using the enzyme PFK1. Thus, it requires fructose 1,6-bisphosphatase to form fructose 6-phosphate. So the fructose 6-phosphate that is formed is converted into glucose 6-phosphate by the enzyme phosphofructoisomerase. So now glucose 6-phosphate is formed from fructose 6-phosphate. Glucose 6-phosphate undergoes hydrolysis by glucose 6-phosphatase, and this also bypasses the irreversible reaction of hexokinase or glucokinase, and it provides energetically favorable pathway for the formation of glucose. The liver is the primary organ of glucose synthesis from glucose 6-phosphate. This process requires a complex of two proteins which are found in gluconeogenic tissue, that is glucose 6-phosphate translocase, which transports glucose 6-phosphate across the endoplasmic reticulum membrane, and glucose 6-phosphatase for the removal of phosphate from glucose 6-phosphate to form free glucose. So till here, these are the reactions of gluconeogenesis, that is pyruvate to oxaloacetate, oxaloacetate to phosphoenol pyruvate undergoing the reversible reaction of glycolysis till it forms fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is then converted into fructose 6-phosphate by the enzyme fructose 1,6-bisphosphatase. This fructose 6-phosphate is then converted into fructose 6-phosphate. This fructose 6-phosphate is then again acted by some enzyme glucose 6-phosphatase to form glucose. So these are the four steps, we can say 4 reactions of gluconeogenesis which are very important. Next, see the regulatory substances of gluconeogenesis. First one is glucagon. Glucagon is that enzyme, or we can say hormone rather, which is active in the fasting stage when the blood glucose level drops. So glucagon is the peptide hormone which is released from the alpha cells of pancreas, and beta cells of pancreas releases insulin. Glucagon lowers fructose 2,6-bisphosphate, inhibit PFK1 and activates fructose 1,6-bisphosphatase, which promotes gluconeogenesis so that free glucose is formed. How is this achieved? Glucagon binds to G protein and it activates adenylyl cyclase. This adenylyl cyclase activates cyclic AMP, which is the second messenger. Second messenger then produces protein kinase A. So the cyclic dependent protein kinase A phosphorylate pyruvate kinase, thus inactivating it. Remember, any kinase enzyme, when it is phosphorylated, it is deactivated. So here also, when cyclic AMP dependent protein kinase A is phosphorylating pyruvate kinase, it is inactivating it. This conversion of PEP, that is phosphoenol pyruvate, to oxaloacetate, that is in the gluconeogenesis pathway, is more favored than the conversion of PEP to pyruvate, that is glycolysis, because pyruvate kinase is phosphorylated, so it is inactive, so it cannot convert phosphoenol pyruvate to pyruvate. Glucagon also increases the transcription of the gene of phosphoenol pyruvate carboxykinase via the transcription factor of cyclic AMP response element binding, that is CREB protein, which increases the availability of the enzyme during fasting. You see, glucagon, it is now binded with the receptor. It is activating adenylase cyclase. This adenylase cyclase is now generating this secondary messenger cyclic AMP. Cyclic AMP activating protein kinase A. Protein kinase A is now phosphorylating the active pyruvate kinase, which is now inactive because it is phosphorylated. Since it is phosphorylated, it cannot convert phosphoenol pyruvate to pyruvate. Thus, pyruvate is available for gluconeogenesis pathway, because pyruvate, you can see, it's converted into oxaloacetate, oxaloacetate to phosphoenol pyruvate, and thus glucose is formed. This is how glucagon regulates gluconeogenesis. Another one is the allosteric activation of acetyl-CoA. So, pyruvate carboxylase, which is carboxylating pyruvate to form oxaloacetate, it is allosterically activated by acetyl-CoA during fasting. Increased triacyl glycerol hydrolysis elevates the formation of free fatty acids during fasting, and these free fatty acids then undergo beta oxidation, which leads to the increase of the acetyl-CoA level. So thus acetyl-CoA now activates pyruvate carboxylase. So you can see here that pyruvate acetyl-CoA is inactivating pyruvate. Because how? Because acetyl-CoA is inhibiting the enzyme pyruvate dehydrogenase complex, that is feedback mechanism, and it, on the other hand, it is activating pyruvate carboxylase. So pyruvate is forming oxaloacetate to glucose. Thank you. I hope this lecture has helped you. If you like the video, please subscribe to this channel and share with your friends. Thank you.